Thank you for the invitation to come here, and now we'll switch to a marine system. But actually, before I will also present the team. We are a pretty large team by now, and lots of the people are actually from Spain and from UK, Sheffield, and then a group in Sweden. I think there is some cutting here with the system, but I will not bother with all these fussy system things. You will now have to live with that we miss a little bit part of the screen. I see already that we are missing 10% in each part, but I think it will manage. Starting with the classical, the textbook example of parallel evolution, of course we all, well, it might be problematic, so sorry, I'll have to get into this stuff that I don't like. Is anyone having a good idea of what should be the display? And then you should show something else here, probably. That one? Mirror display. Okay. Yeah, thank you. This classical system, as we heard this morning, may not be so classical anymore, but still I will use it as a kind of introduction here to make the concepts all right. I mean, what we say when we mean parallel evolution is, of course, that we have a phylogeny that will look like this one. So even if you have two ecotypes in the same lake, and they may be quite much similar between lakes within ecotypes, we still expect that they have evolved repeatedly and independent of each other in each lake. However, it's quite easy to come up with some alternatives that will eventually look quite similar if you do a phylogeny. And one example is, of course, a secondary invasion, a double invasion. That is, you have an allopatric separation and origin of the two ecotypes, and they come together. And if they in the same lake then start to hybridize extensively, of course, after a while, you can have a pattern which looks quite similar to a parallel evolution pattern. We tried this once to discriminate between these two possibilities, and it can be done, and I refer to this publication by Panova et al. If you're interested in that. But I will take this a bit further and a little bit consider what is the problem with parallel evolution, and to be really sure that you have parallel evolution, or that if you have something else. Well, of course, first, if we think about the very simple scenario where we have completely independent sites, two lakes that are having no gene flow whatsoever between them. And if we also can go back in time and say that even during the recent history, these lakes have been separated for a long while. Of course, there is no more possibilities than these two ecotypes that are very similar but live in different, the populations of the same ecotype living in different lakes. They must have evolved by something we may call parallel evolution, or we may call convergence. That was actually suggested by Arendt and Resnick, because there is actually no big difference between parallel evolution and convergent evolution. It's a matter of, the same process. So we can use the name convergence, because then we know what we are talking about. However, if we have an ancestral situation where these two lakes were not independent, they will be quite likely to share a lot of variation. And of course then we may expect that this evolution of the local ecotypes may be from the shared pool of genetic variation. And then we have no parallel evolution, not in the strict sense. We have no convergence. but we have evolution from a pool of shared standing genetic variation or shared genetic variation. So that is a completely different scenario. But still we will end up with the same pattern of the genetic structure, because we cannot really say that, or we cannot distinguish this pattern from a classical phylogeny based on parallel evolution. Even more complicated will be when we have a situation where we still have some gene flow, even if the gene flow is very tiny. It may be so tiny, so the large part of the genomes, the neutral parts of the genomes, will not be affected. But still, alleles or new mutations that are under strong selection may be transported quite a while and over quite long distances, and occasionally between fairly isolated lakes or populations. And then, in this case, we will have a similar genetic structure that looks very similar to parallel evolution. Although in this case we have indeed not full independence between the two lakes. So there are quite many different alternatives to what we have been calling parallel evolution. The allopathic origin, the double invasion, the convergence, evolution from standing genetic variation, and then this last possibility of evolution in concert, as we have been calling it this. So now we'll switch to a system which is quite like the stickleback system overall. We have different areas, different sites, and in these sites we find very different ecotypes. So if I show if I show a range of ecotypes, as in this picture, you will almost not believe me when I say there are ecotypes of the same species. So these are all adult individuals of Littorina saxatilis. Yeah, I try to press the green, but, well, anyhow, you see this small tiny one up to the left. There is an adult living inside empty barnacles, and you see the big one below. That is an adult form that you found in northern countries. To the right you have the ones that we have been interested in this particular study that we are, or in a number of studies that we have been performing. There are the Spanish ecotypes and the Swedish ones, and I will show you later on also the British ecotypes. But all these ecotypes, they tend to be locally adapted over a quite small geographic scale. So for a Swedish system, for example, you can find within the same bay of an island, you can find both the wave ecotype, and you can find 30 or 50 meters from there the crab ecotype. And in between you can also find a zone where you find a few hybrids. And these hybrids are fully fertile. But still we can measure a reduction in the gene flow between these. So there is a very clear reduction in gene flow, which we can measure using microsatellites, for example. The background to the ecotype formation is, of course, that there is a lot of selection in different directions here. So in the wave-exposed cliff side, we have wave ecotype surviving about 10 times as good as the crab ecotype, and the other way around in the boulder beach where we have lots of crabs, the wave ecotypes are doing quite bad, while the crab ecotype is doing quite well. And also, in addition, I should say that they have a direct development, so there is very little dispersal as a whole. They live within the same square meter, or more or less, over their whole lifetime, these snails. We know from a lot of experimental work that they also have quite an important degree of associative mating. So if you put them together in an aquarium, the male actually chose, in most cases, to mate with a female of his own type. And a part of this associative mating is actually based on following the mucus trail of the female. So already at the stage of crossing a mucus trail, the male can decide if it's a female of his own type or of another type. And altogether, you can actually say that this associative mating at different steps will account for all the decreasing gene flow that we can see in nature. So we can explain the decrease in gene flow over the hybrid zone only by the amount of associative mating that takes place. Okay, so now we have 3 different systems. We have the Swedish system, where we sampled 2 sites, the UK system and the Spanish system, and I will now present some ongoing work. So I should say that a lot of these analyses are preliminary, but still I think they are indicating some interesting patterns here. And in all these sites we have both the crab and the wave ecotypes, and of course it's a bit of a first issue. Are they actually similar? Are they the same ecotypes or not? So what we did as a first thing was to study the morphology using classical morphometry. And what you can see is that in the first place here we have actually from the 2 different sites within the same country, we have consistent patterns of wave ecotypes and crab ecotypes, and they are split so much that there is practically no overlap. But also between countries the patterns are the same, even if there tend to be that the Swedish ones are perhaps a bit more separated than the British ones, but there is in principle the same separation. So we can surely say that we have 2 different ecotypes around all over the European continent. Then we were interested, of course, to know what was the level of independence here. And the first thing to do, of course, was to try to analyze the phylogeny using a mitochondrial marker. And of course, this is only one marker, and as you will see shortly, there is fairly different patterns between the different markers that we used. So when we compare them with a nuclear marker, we get a bit different patterns. But for the mitochondrial DNA, it's quite interesting to see that the Swedish and British sites or populations are not separated. They are not separated in geography, and then they are not separated in ecotype, so they're not independent. But in Spain, on the other hand, we seem to have 2 different areas. So the 2 Spanish sites are really independent from each other, because you have the haplotypes from one area here, and then the haplotypes from the other area here. So they are 2 independent groups. Unfortunately, the nuclear genes came up with 2 different patterns, so these are preliminary analyses, but for one of the genes we have so far, we plan to have at least 3 genes eventually, possibly more. We have no separation at all, so we have no independence. And for the other gene, we have all the countries actually independent. So we now have 3 different suggestions of independence. And of course, this is not unexpected, because gene trees tend to be different. And of course, to go for the, well, to look for the species tree, we must integrate more information here. So either we have this type with 2 independent Spanish sites and the other ones grouping together, or we have no independence at all, or we have 3 independent sites. Using another marker or another approach, we looked for the genetic structure with the AFLP, and of course that is, the promise of that is that you can cover much more of the complete genome. And in this case, we can see that there is actually a very nice structure distinguishing the three countries. There's also structure within countries. So in this case, we have a complete grouping which is based on the geography, nothing else. And the two ecotypes, they don't turn out to be a base for grouping whatsoever at this level. More interestingly is perhaps to do an outlier analysis on these data and to separate out the loci that may be on the selection and see how they group. Well, doing this, we can first compare within a country and do an outlier analysis for one site. And for one site then, we will find 57 candidate loci that are diagnostic in the way that they separate the crab and the wave ecotype. So they may explain the evolution of the different ecotypes in that site. In another Swedish site, we've found 30 of these. But interestingly enough, only 5 of these were shared between the two Swedish sites. And similar for UK, we only had 1 candidate that were shared, and in Spain only 3. So there were quite a few of the genes that were outliers that actually were shared among the sites, showing that they were contributing to the evolution of the different ecotypes. And if we looked throughout all the sites, we actually found no gene that were consistently pointing out a difference between the ecotypes. So at this level, with this information, we have very little support actually for the same alleles or the same mutations contributing to the evolution of ecotypes. Rather, this supports what can be something like parallel or convergent evolution. Also, if we use only the information from the outlier loci, it's really hard to see the pattern that separate the ecotypes from each other. So here in this cluster analysis, you can see that Spain is still grouped separately from the other sites. And in UK, for example, we have a geographic grouping rather than an ecotype-based grouping. So still weak support for standing genetic variation or any of the non-convergence ideas. Finally, we also have started on a microarray approach, and with such an approach and covering an extensive part of the complete genome, more or less more than 25,000 gene models, we did a first very simple analysis which is called Comparative Genomic Hybridization. You simply test how much of genomes from separate individual snails will be able to hybridize on the microarray. And doing this, we had a nice separation throughout the countries. However, if we look at the separation again, of these 26,000 gene models, we have about 1,500 that separates out the 3 countries, as was shown in the picture before. Quite few of these actually separate Sweden and UK, only 8 of them. But there is much larger separation between Spain and the other countries. But if we go to the crab wave ecotype separation, there is very little to be found actually. Only 1 gene model out of 26,000 indicated a separation between the ecotypes. And in UK and Sweden, we actually didn't find any gene model separating the ecotypes, while in Spain, we found 27 out of 26,000. Of course, here we can be a bit relying on the fact that this genomic hybridization may not be so sensitive. So it may not be able to pick up single mutations. And this may be the explanation for the difference between UK, Sweden on the one hand and Spain on the other hand, that Spain is probably an older system. It has a history which is older than since the last glaciation, probably. So the preliminary conclusions at this stage is that we have populations that seem to be, at least to some part, independent between countries at least, although one of the nuclear markers didn't indicate this. So there is also, of course, a large part of shared ancestral variation. And although we have many loci that separate between the two different ecotypes, there are very few consistent separations over this scale, this geographic scale. So we have a strong support at this stage that at least on the large geographic scale, we may have some parallel or convergent evolution. And to some surprise, also it seemed to be that also within countries, this can be true. Now, I should finally like to actually give an example from a related species, which we have been studying or are presently studying, in which we actually find a very, well, probable example of evolution in concert. That is the fourth mechanism that I mentioned. This species is Littorina fabalis, and it has quite a similar system as for Littorina saxatilis, so you can find different ecotypes. And the two most common ecotypes is a dwarf ecotype, the SS ecotype, and a large or a normal size ecotype, the LM ecotype. And both these ecotypes are found all over the northwest Europe. So over UK and Sweden and Norway you can find, and along the coast of France, you can find these two ecotypes side by side. As well as for Littorina saxatilis, they occupy slightly different microhabitats of the shore of the same base. And we have been looking at the allozyme system for one of the allozymes called arginine kinase. Well, many years back, we actually found that in this allozyme there were a very strong correlation between frequency of one allele and one of the morphs and the frequency of another allele and the other morph. And now I had a master student who actually sequenced these alleles, and first he sequenced the introns, and here he used samples from Sweden. They are flagged in blue and yellow as a Swedish flag, and then he used samples from Wales. And I think this has something to do with a football team in Wales or something. There are green and white colors, or it might be the flag. But what you can see here is that one of the ecotypes, the LM ecotype, which is also marked in black, has several haplotypes present. So in this post-glacial system, which is about 10,000 years old, we have several haplotypes in one of the ecotype. While in the other ecotype, we have only one haplotype present, both in Sweden and in Wales. And now this is the intron part. It's the part that is not used by evolution. So it was a bit interesting to see if this will also be the case for the exon part, which might be under selection. And indeed, going to the exon part, the situation is even more extreme. We still have more haplotypes in the one ecotype, and we have only one haplotype in the other ecotype. And these are actually separated by a number of non-synonymous mutations. So the interpretation of this is that this gene and the haplotype or the allele that is represented in one of the ecotypes here has actually a very recent origin, and it has spread over the complete area, probably quite recently, while the LM ecotype is the carrier of the ancestral part or the ancestral allele, and therefore also this allele hosts more variation. And it's interestingly that the differences here are, well, at least suggesting that the new allele here is under heavy selection, because otherwise you will not expect that many number of non-synonymous mutations. So, trying to sum up here, we have quite a complex picture now. We can probably, at least for the genome as a whole, actually reject the idea that we have an allopathic origin of these ecotypes. But, of course, we still have the possibility that single genes have an allopathic origin, and, for example, has spread by selective sweeps or is present in the standing genetic variation. But this seems not to be the story for the genome or the majority of the genome. Rather, we have indications that parallel evolution, or preferably convergent evolution, is quite likely, at least on a geographic scale, and possibly also on a medium scale, that is within countries. For standing genetic variation, we have already evidence from back in time when the allozyme studies were performed, because there were plenty of examples where alleles were at low frequencies in some habitats and then increased in frequency whenever they were under selective pressure. So yes, we have standing genetic variation for sure. And that might be the more important part for parallel evolution at the smallest geographic scale. And then finally, we have at least one example, while in Littorina fabalis, of a system which seemed to have evolved in concert with a very successful mutation spreading over a large area and coming into use in the formation of one of the ecotypes. So the picture is complex, and I think still we need some more time, of course, and some more effort and a lot of more money to get the full elephant out of our work. We have only parts of it yet. So thank you very much.
Thank you. We have a question on the third row.
Well, I enjoyed very much your talk, because you are dealing with a very, very essential problem in evolution, you know. But I think it's important to distinguish two things here for people. One is sympatric or parapathetic speciation. The idea is that, yes, sympatric or parapathetic speciation is shown by your examples. Maybe you can comment on this. And that means that natural selection is operating in this kind of speciation. Okay? But the second point is, what do we understand by parallel evolution? Because this has been one of the more contentious things, the most contentious thing in the whole history of evolution, you know. If you know, convergence has been also demonized by many people, like especially people like Steve Gould, that he didn't think that convergence was important, and because there is the constraints and all the ancient developmental genes or factors that canalized the possible issues in evolution. So when you are talking about parallel evolution, what are you talking about? You're talking about this concept of some developmental geneticists or some developmental evolutionists that they like Sean Carroll, for instance, that they are trying to say that it's more correct to talk about parallel evolution than convergence, in the sense that there is a lot of ancient homologies, you know, that you always have there, but you didn't detect because those are not in the coding genome, but they are in the regulatory part of the genome, and now we are starting to unveil these things. And in parentheses now, maybe if you look at this part of the genome, maybe if you look at this part of the genome, just comment on this, you could find more parallelism maybe than looking at the coding part. So what do you think about this?
I can start with the issue on sympatric and allopatric. I mean, of course, I mean, we can claim that today we have more or less parapatric scenario. I mean, they're not living on the same square meter, so there is a gradient, and that this gradient is quite important. But of course, as I try to emphasize in my talk, it's really very hard to trace the origin of the genes that are important for the ecotype formation. So some of these genes may actually evolve independent of each other in different places, and they will then spread over the complete distribution. Others may evolve locally, or they may evolve in parallel. So I mean, there is a whole bunch of different possibilities creating the genetic variation that is then used for evolution of the ecotypes. Given that this variation is already there, I think that this example we have here is a very nice example on parallel evolution. But then you can actually say that this is parallel phenotypic evolution. It's not parallel evolution in the sense that were originally described. I mean, where you expect that you have the same substitutions, the same mutations coming in independently different places. Because if you have standing genetic variation, you obviously don't have this. You may have just one single mutation that has first spread, and then you have this variation everywhere, and you can use it. So I think we have to be clear on what we mean by parallel evolution in that sense. And I think this is perhaps part of the answer to your second question as well. I think that what we are now going to need is to actually go into these candidate loci that we know are important for ecotype formation, and then we need to sequence them. We need to sequence the intron parts and the exon parts to try to understand their background. Do they have one origin, or do they have several origins? That is the first question.
I think we will have to see if there are other questions. We can take one more. So a taxonomic rather than an evolutionary question. What has happened to the taxonomy of this Lewis group? Because there was Littorina, saxatilis, neglected, and also are we talking about the same thing as Maria and the other species you referred to?
Well, neglecta was the small one, so it's not now an ecotype. But going back in history, I can say that this species, Littorina saxatilis, which is now considered a species, was described under 28 different species names. And I mean, it's not surprising. If you go for the morphology here, you cannot see what's behind it. Then is, of course, another issue. Are these species, these ecotypes? They are separated to 90%. So of course we can give them species names. But should we then give a separate name to each island in the Swedish archipelago? Then we will increase the biodiversity by, I don't know how many times. So.
All right. So let's then just